Hi, everybody. Conboy here. You and I have an ambitious objective to meet, and that is to kick off the DNA structure function part of our unit by getting an overview of a field of research that is amazingly wide and incredibly deep at the same time. That is DNA research. It consumes billions of dollars per year. It is massive already, and it is growing at an exponential rate. In this presentation, I'm going to take a fair amount of time to explain some concepts. When I do, take good notes. If I spend more time explaining it, that means it's extra important and will be on the test. Other ideas, I might just refer to the diagram and encourage you to study it on your own just for the sake of interest. But you may not have to know that for the test. So keep that in mind as we go through this, because sometimes I'll be careful and sometimes I'll go fast. Now, if you're going to understand why DNA matters, you first have to know why a cell matters. Now, I know you all know that your body is made of trillions of cells and that those cells somehow cooperate, but I don't think everybody knows what each cell is responsible for doing. Essentially, you should think of a cell as like a factory. It's responsible for manufacturing a product from scratch. Now, cells, of course, are very tiny, so its products are all on the molecular level, pretty darn small. Now, if you want to understand a cell's basic job, then you should think about a factory's basic job and what that factory needs to succeed. That will help you remember how DNA fits into the cell. Let me show you. Of course, any factory, say one that's in charge of building cars, needs a power supply. In a cell, the power supply is ATP, which is a special type of molecule which we'll study in great depth later. But for now, you should just know that every cell ever studied in the history of biology uses ATP to power its activities. And in fact, viruses do not make ATP, they do not use ATP, and those are some of the reasons they're not considered alive. All factories have to use some kind of raw materials. To manufacture a car, you'd have to have a variety of metals and foams and rubber or plastic. In a cell, most commonly, we need amino acids. And amino acids, you might recall, we can get from our food by eating protein. Of course, a factory needs a variety of workers and a location to do their work, called the assembly line. Ribosomes provide both to the cell. Now, it would be pointless to spend lots of power and raw materials building something in a factory unless we had good instructions to guide us. In the cell, those instructions are provided by DNA and a similar genetic language called RNA. So, in order to understand anything in the rest of this presentation, you have to first understand that DNA and RNA are giving the cell guidance in how to build whatever it's supposed to build. So, the cell has the same basic needs as a factory. I expect that you would understand the basics of a factory because our economy is so industrialized, and that can help you remember what's up with cells. So, what do cells even build? Well, fundamentally, the product they make, not a car, but a protein. They make proteins. Now, proteins, of course, you eat in your diet. Why don't we just take the protein from our diet and just use that? Well, that's because the protein in a steak, which was made by a cow, may not suit the need of the human. So, what we have to do is take our digestive system and depolymerize the proteins we eat into the individual monomers called amino acids. So, that digestive process then produces for your cells the raw materials or amino acids that are then recombined into new polymers, long chains of amino acids that will suit the needs of the human. Now, how do we know what amino acids to combine and in what order? Well, that's all programmed by DNA, which is why DNA matters so much. Now, we should say a little bit about amino acids before we go too far. So the products that DNA have to make are made of amino acids. So let's think about what each amino acid has. There are many types of amino acids out there, but there are really only 20 that are commonly found in proteins. You don't have to memorize those 20. That would be hell. Trust me, I've been there. Every amino acid has in common two functional groups. One of them is called an amine group or amino group, NH2. The other functional group we always expect to find is a carboxyl group, C double bonded to O and to OH. Now, hopefully, you recall that we should be looking for OH groups. OH groups are commonly involved in forming bonds between monomers to make polymers. So here we have two different amino acids side by side. I'm hoping that you could see that the OH from leucine's carboxyl group could potentially interact with this H from serene's amino group to form a covalent bond. And that would connect leucine and serine together, starting to build a polymer. And in the process, we would lose this H and this OH in the form of a water molecule. That's what we call a condensation reaction or a dehydration reaction. We've seen that before. So these amino acids connect side by side between the carboxyl group of one and the amine group of the other. But what's really interesting are the side groups. So the side group is what makes each type of amino acid different from the others. And the side group is always sticking out of the carbon that's in the center of the amino acid. So leucine has a side group that's very different from serene's. Leucine's side group is going to have all nonpolar bonds, meaning it can participate in boring IMFs. The boring IMFs are London dispersion forces. But serine, on the other hand, can participate in IMFs like dipole-dipole or perhaps hydrogen bonds. And that's because it has a polar hydroxyl group there. So what you need to imagine for now, and we'll practice later, is that a long strand of connected amino acids would put lots of different side groups near one another. And those side groups could then attract or repel from one another in funky ways based on IMFs. And so what is really a long polymer could fold up in all sorts of crazy ways, depending on what amino acid is placed where. Of course, what amino acid is placed where is based in DNA. But the shape of proteins essentially is based on the sequence of amino acids and their functional groups or their side groups. Now, there are three types of proteins. I'm going to say a little bit about each, and we're going to study them at different points in the year. We have structural proteins. These are proteins basically that build up your body. And, they might not have much of an obvious function, but they're often asked about in the genetics unit we do. So, for example, at some point you guys are going to ask us, hey, why do our skin colors vary? Why does my hair color differ from that of my brother? Or why do we have different eye colors in my family? Well, we can blame all those structural proteins on differences in DNA. We also have a special type of proteins called hormones. And these are proteins that have a role in a message getting from one cell to another. For example, adrenaline is a protein made of amino acids that is made by some cells in your body and then delivered to other cells in the body to get them to react to a crisis situation. So if the job of the protein is just communication, then it's a hormone. And then, of course, we've recently talked about enzymes, which are a specific type of protein that have the job of simply catalyzing chemical reactions. Now, no matter what type of protein we're talking about, the cell that makes it can do one of two things with it. It can keep that protein right in the cell where it was built, and it can be used there to perhaps repair some other protein in the cell that's broken, that needs to be replaced, or it can build up a supply of them so that if the cell reproduces, the offspring cells start off with lots of protein. The other thing is that a cell can make a protein and have the job of sharing it with other cells in the body. So going back to adrenaline, if the adrenal glands sitting on top of your kidneys make the adrenaline on the inside of their cells, If those proteins, if those adrenaline molecules have to get to the brain and the heart, then that means that the proteins have to be exported out of the cell. So, a common way of exporting proteins, let's say these blue things are proteins, a common way of exporting proteins is to package them inside a vesicle within the cell and then send that toward the cell membrane or the plasma membrane. Then exocytosis will basically turn this vesicle inside out and dump the protein outside the cell. Now, of course, in a big animal like us, there's a bloodstream that can pick up those proteins, and the blood will move them away from that cell and maybe towards cells in the heart or the lungs so that they can perform extra well.